In these lessons, I will attempt to penetrate your mind. You will attempt to resist. Prepare yourself.
Alright, this video is going to be over the lab and lab safety as well as the scientific method, so we'll go ahead and jump right in. This is the scientific method that you've seen probably in every science class you've ever taken. Okay, we start with our question up top, and we go on, we do some observations and some research about whatever it is our question was revolving. Then we move on to our hypothesis. Remember, you have to have two things to make a good hypothesis. The first one is that it must be testable. Remember, that is confined by whatever you have in your lab and have readily available to you at the time. And secondly, it must be conditional. And you know it's conditional if you have an if and then statement. Then you move on through your experiment, you analyze your data, then you decide if your hypothesis is supported or refuted. If it's refuted, you come back up, think of a new hypothesis, and start the whole process over again until your hypothesis is supported. Once it's supported, you move on to your results and you report those in some kind of peer journal or lab report. So go ahead and pause this and do these questions over the case of Berry, as well as deciding if those three hypotheses are good hypotheses or not. Okay, let's move on to different types of data. The first type of data you have is qualitative. And qualitative is based on descriptions, so you have examples like color and clarity and texture. A good way to remember these is that these are non-numerical observations. The ones that have numbers, those are called quantitative measurements. Anything that has a number associated with it, such as mass or volume or length, those are going to be your quantitative observation. So go ahead and pause it again and go through your practice questions and see if you can decide if they are quantitative or qualitative. Okay, now let's talk about laws and theories. Let's talk about laws first. The first thing you need to know is that laws are concise and factual statements that's meant to prove some kind of observation. The laws are also accepted to be true. They're accepted as universal by scientists all over, and it's often expressed as a mathematical equation. So you can go ahead and think of some laws and go ahead and write those in your notes. Theories, on the other hand, are explanations of a natural or physical phenomenon based on multiple hypotheses, and that's your key there. Multiple hypotheses, they're verified multiple times by multiple researchers. Theories often include laws to prove their point. We'll talk about those in class. So your variables. Every time you're talking about variables, you have three different kinds you need to be aware of. The first one is the independent variable. This is the variable that you or the scientist is changing. It's going to actually affect the dependent variable. When you turn these into a graphical representation, you do that on the x-axis. Dependent variables, on the other hand, they're measured in response to the change of the dependent variable. It's what you're actually recording, it's what you're actually testing. When you graph these, they go on the y-axis. And then you have control variables. Keep in mind, these are different than control groups. They're held constant. These are going to be things like temperature of the room, humidity of the room, things like that. So go ahead and pause it and go through the vacuum cleaner. Okay, now let's talk about the differences between mistakes and sources of error. When you make a mistake, it's generally some kind of procedural mistake. You didn't follow procedure the right way, you did something in the wrong order, you didn't add in enough, or you didn't take out enough of whatever it is you're working. Sources of error, on the other hand, are limitations to that procedure. So these are things that, could you have done the procedure more precisely? Could you have used a better, more precise instrument? Is there something that's contaminated? Maybe whichever you're working with is contaminated. Then make sure your control variables are actually held constant. If they're not, you can always go back and see if you can control that depending on the equipment you have. And then all your assumptions. Those are all sources of error. Remember, anything that limits the procedure is going to be a source of error. Any type of procedural mistakes that you made during the lab are going to be mistakes. So go ahead and see how you can do on these. These might take some practice until it starts to click, but go ahead and pause it. Alright, let's get into some lab safety. The first thing you should already know is you're going to wear goggles at all times. You're also going to wear closed-toed shoes at all times. That's to prevent chemicals from getting on your feet, as well as they'll protect your feet if in case any glassware gets broken. You're also going to wear an apron during all labs. That's to protect your clothing. Eating and drinking, and that includes gum chewing. It's never going to be allowed during the lab. You're always going to wash your hands before leaving. That's just to make sure you don't spread any chemicals to any of your friends outside of the lab. And then if you've got long hair, you're going to pull that into a ponytail or put it up somehow. That's to keep it from getting into the chemicals themselves. And also for working with fire, we really don't want your hair to catch on fire. For anything that produces a toxic gas, and that's the fume hood, right over here. You should have one in the back of your lab. And while you're working with chemicals, a lot of times we'll have a big jar of stock chemicals, and you're going to put those stock chemicals into smaller jars. If you take too much, don't ever put anything back into the stock container, because that will contaminate the rest of the stock container. If you're disposing of chemicals, always ask the teacher how you want to do that. Generally, we'll tell you that we'll dispose of them. Don't just take a hearsay from your lab partner about how to dispose of them. When we smell chemicals, you're going to do it by wafting. You can see this guy right here in the lab coat is doing the wafting. That's how you do it. Don't put your nose right over your test tube or your beaker, because sometimes the fumes can be so strong that they might knock you out. Then anytime we have pointed test tubes, you need to point those heated test tubes away from other people and yourselves. Things tend to shoot out of those, and we don't want that hitting you or somebody else. And the big one is when you're mixing acids and bases and water, you're always going to add the acid into the water, never the other way around. That's to prevent splashing. If you see an event of emergency, you're always going to tell the teacher. We'll know how to handle those situations. If you're working with a spill, if you spill an acid, you're going to use a base to neutralize it. And our base that we're going to use during the lab is baking soda. And the opposite's also going to be true. If you spill a base, we're going to add an acid to that, and our acid that we're going to use is vinegar. It's acetic acid, and that'll neutralize any basic spills. If you get a chemical in your eye, which shouldn't be an issue, you're going to use the eye wash station, and that's what this picture is right here. You have water squirted into your eye for 15 to 20 minutes, and that's just not fun, so make sure you're wearing your goggles at all times. And if you get them on your body, you're going to use that safety shower. That's what this is over here. That should get the chemicals off your body. In the event a small fire starts, you can always take a beaker and invert it on top of that fire. That'll suffocate the oxygen out of that flame, and that'll put it out. But if that fire catches on somebody, you can either use the fire blanket or the safety shower. Whatever's closest to you will work. Vocabulary that you're going to see when you're working with chemicals, you need to be able to tell us what that vocabulary means. So the first one, if you see the word corrosive, that's going to be something that's going to eat away at objects. It's going to burn your skin. Anything labeled as toxic will be poisonous. Inflammable, obviously, going to be set on fire, combustible. Carcinogens, which you guys might be familiar with, those will cause cancer. Radioactive things, we're not really going to work with those in the high school lab, but that's anything that's going to emit radiation. And then you have allergens or irritants, and that can cause rashes or allergic reactions on your skin or in your eyes. Now let's talk about these safety diamonds. You're going to see these safety diamonds on a couple of different stock vials of chemicals. You're going to see them in colors, just like this. The blue, if you pause it, and if you want to go back and take a look at it, the blue is going to be health hazard, red is flammability, yellow is reactivity, and white is going to be any special information that you need to be aware of with the chemical. And each one of those colors is going to have a number associated with it. You need to know what those numbers mean. They go from lowest intensity to highest intensity. A 0 is going to be no hazard, a 1 is a slight hazard, 2 is hazardous. And once you get to 3s and 4s, we don't really work with those in the lab, but 3 is extreme hazard and 4 is deadly. Now the white, you're going to have special symbols on it. In this one, in case the water, you see the W with the line pushed through it, that means don't add water to this substance. In any event you want to know more about the chemicals, you're going to have these MSDS forms. These are material safety and data sheets. This one's for hydrochloric acid. It tells you just about everything you want on it. It'll tell you the different types of hazards. In the case of an emergency, it has these first aid measures down here. It has things involving flash points and how hot it has to be to set on fire. Anything you could possibly want to know about the chemical you're working with is going to be on this MSDS sheet.
Are you going to teach us anything, or are we just going to sit here?
Just do whatever you want.